Hi, everybody. Okay, welcome to your part 2 of chapter 15 video. And just another quick reminder that you do have a homework that is due. If you're watching this in the fall of 2020, you have a homework that is due on October 23rd, which is Friday, and it does cover chapter 15, which is the one we're working on now, as well as chapter 16. Okay, so when I left you at the end of the part 1 video, this was the slide we were on, and I just kind of want to recap really quickly what's on this slide, because if you understand this, it's going to help you to understand how DNA is replicated. So remember that DNA is, of course, a double-stranded molecule. And of course, what Watson and Crick were very famous for discovering was that this double-stranded molecule was, of course, a double helix. So they discovered that it had two strands, They discovered that it had two strands, that they were wound around each other in a helix, and that the bases pointed toward the interior and followed complementary base pairing rules where A pairs with T and C pairs with G. Now, if we untwist this to look at a simplified model that looks kind of like a ladder, I want you to think of the sides of the ladder as the sugar phosphate backbone. So if you recall from our previous lecture, here's what that would look like. So here's our sugar attached to a phosphate. Oops, let me forget, there's another carbon there. Here's a phosphate. Don't let me forget, there's a carbon there. Oop. And there's a sugar attached to a phosphate, etc. And then there would be an open OH group down here. So a couple things I want to point out. This red sidebar here is intended to represent a sugar phosphate backbone like what you see here, okay? These sugars and phosphates are welded to each other by covalent bonds. Remember, from way back at the beginning of our class, these covalent bonds, they are permanent and they are strong. So all of that is to say that this sugar phosphate backbone is not going to fall apart very easily. It's going to stick together. Another thing to notice about our sugar phosphate backbone is that it's oriented with a phosphate group on one end and an OH group on the other. And remember that the phosphate group is attached to the 5' carbon on our sugar molecule, while our OH group is attached to our 3' carbon on a sugar molecule. So we would say that this particular sugar phosphate backbone goes from the 5' to the 3' direction, which is what we see here, 5' to 3'. So you could think of this end as having a phosphate group, and this end is having an OH group. Now recall that this OH group is really important because you can probably tell that if we were to add another nucleotide to this strand, it would be added on at this end where the OH group is. So we would add another one on there or another one on here. And that's going to be really important in a little while when we start talking about how we replicate DNA. All right, another quick review. Remember that DNA is a double-stranded molecule, so this is one strand here. Let's look at its sister strand here. Notice that its sugar phosphate backbone is upside down. Here's our 5' end, where the phosphate group would be poking out, and here's our 3' end, where we would have our OH group. And so these are anti-parallel molecules. They are upside down relative to one another. Now, I mentioned that our sugar phosphate backbones are really well stuck together. They're welded together by covalent chemical bonds. But let's look at the interior of the molecule here. What's happening is we have these complementary base pairs. Here's an A and a T, a C and a G. And what is holding these complementary base pairs together? We know what's holding the backbone together. That's a covalent bond. But how are these rungs of our DNA ladder, how are they sticking together? And if you remember from our last lecture, they are being held together by hydrogen bonds. And hopefully you remember from earlier in the semester that hydrogen bonds result from molecules that have partially positive charges on them interacting with molecules that have partial negative charges on them. So they stick together kind of like magnets. And just like magnets can stick together and pull apart, so can these base pairs. So what that means is we could pull this strand in this direction and we could pull this strand in this direction. So let me just erase a little bit here. So what I'm suggesting to you is that if we pulled these two apart, here's what it might look like, where you would have one strand here and one strand here. Now, this kind of immediately suggests how we might copy our DNA to make new copies. Because if we pull these things apart, if we can just get a hold of some nucleotides that might be floating around inside the nucleus, then these nucleotides, these individual nucleotides, could line up on these strands according to complementary base pair rules. And all we would really need is an enzyme that could come along and weld together the covalent bonds on that sugar phosphate backbone. So let's do that again. Here we can have some individual nucleotides that could come along and pair up according to complementary base pair rules. And we just need for an enzyme to sort of zip up that sugar phosphate backbone. Now that's an oversimplification of what happens, but essentially you can think about this as how DNA becomes replicated. Now in 1953, let me erase all this because this was from an earlier recording that I ditched because I messed something up. So let's erase all of this. So in 1953, when Watson and Crick published their paper describing the structure of DNA, they knew that this structure immediately suggested how DNA might be copied. But there was a bit of a problem, and that problem was that we did not have any experimental evidence to demonstrate how DNA was copied. And so there were really three hypotheses for how DNA might get copied. And we're going to go ahead and talk about each of these individually, but the three hypotheses were the semi-conservative replication hypothesis, the conservative replication hypothesis, and the dispersive replication hypothesis. Okay? So here is what each of these hypotheses entails. So let's take a parent strand of DNA, and I'm just going to draw it in black and draw it like a ladder. So here we go. And then here's its sister strand. And remember, these are being held together by a hydrogen bond. What the semi-conservative hypothesis of replication does is suggests that these strands pull apart and each individual old strand serves as a template to make a new strand. And so let me draw a new strand here in orange. Here's a new strand here. Boop, 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 boop. And here's a new strand here. And so we call this semi-conservative because in this strand, in these two strands, these old strands, sorry, in these two new molecules of DNA, these old strands were conserved while we have new strands here. So we have a semi-new situation, right? It's kind of half and half. Let's contrast that with this idea of conservative replication. In conservative replication, what folks thought might happen was that you had an original strand of DNA and that somehow this entire strand was copied like a photocopy so that you'd have one entirely old strand and one entirely new strand, okay? And we weren't exactly clear on how we thought this might happen, although some folks suggested that somehow perhaps maybe this molecule turned inside out so that these bases were kind of poking out and could be copied, okay? So you can see why this is called conservative, because here we have conserved our old strand entirely of DNA, our old molecule of DNA, and then we've made an entirely new molecule of DNA here. The last hypothesis for how DNA might be replicated was called the dispersive replication hypothesis. And what this hypothesis suggested was that you would have an original molecule of DNA here, and that somehow this molecule might be cut up and copied so that your resulting, let's see, how do I want to do this? Your new molecules of DNA might have some old parts and some new parts and kind of be like a chimera of old and new together, okay? Let me just kind of draw this out for you. And again, it wasn't entirely clear how this might happen, but these were three thoughts on how you might end up with copied DNA. Now, as I mentioned, the problem here was, while most folks, Watson and Crick included, believed that the semi-conservative hypothesis of replication was the most likely way that DNA was being copied, we didn't have any experimental evidence to demonstrate this for us. So in 1957/58, sometime in this time frame, there were two scientists that were working, and they were friends of Hershey and Chase, who did that first experiment we talked about earlier in the chapter that definitively showed that DNA was the hereditary material. So these guys were friends with them. But these two scientists are Meselson and Stahl, and this is a very famous experiment called the Meselson-Stahl experiment, okay? Now notice, this happened in 1957, 1958. So if you're keeping up with the timeline, in 1952, we have the Hershey-Chase experiment that really shows us that DNA is the hereditary material. In 1953, we have Watson and Crick showing us the structure of DNA. And here we are in 1957, we're going to learn about, let's put MS for Meselson-Stahl, and we're going to learn about how DNA is copied. So hopefully you can see that this was a very rapid time of development in our understanding of DNA. And the 1950s really ushered in what we consider the modern era of genetics. Now, what Meselson and Stahl decided to do was to take advantage of the fact that DNA has tons of nitrogen in it. And if you think about what like the structure of a nucleotide is, there's our phosphate, carbon, here's a sugar, an OH, and then remember that we have the bases here. These bases are often called nitrogenous bases. They are full of nitrogen, okay? And the thing about nitrogen is that there are a couple of different isotopes of nitrogen. Normal nitrogen has 7 protons and 7 neutrons. And so we call this N14. This is a normal nitrogen atom that you would find in a normal molecule of DNA. But if you remember from earlier in our discussion about atoms in this course, there are things called isotopes where they have the same number of protons. So here's a nitrogen. It's going to have the same number of protons, 7. But this nitrogen has an extra neutron. It's got 8 neutrons. This is called N15, and it's radioactive, and it's also sometimes called heavy nitrogen because it literally has more mass than this guy does. If you had 100 atoms of N14 and you could weigh those, and you had 100 atoms of N15 and you could weigh those, this sample would be heavier. So here's what Meselson and Stahl did. They took a bunch of bacteria, our good friend E. coli, which you've heard of before, and they grew it up in heavy nitrogen, okay? That means that as our bacteria are growing and making DNA, they are using heavy nitrogen to make their DNA. And so if we were to take these bacteria and we were to put them into a test tube, okay, and then centrifuge that test tube, hopefully you know that when you centrifuge things by spinning them around and around and around, what happens is it slings heavy stuff to the bottom of the test tube and light stuff stays toward the surface. So anything that had heavy nitrogen in it would sink to the bottom of a test tube. If you had something that had lighter nitrogen in it, it would be maybe toward the middle of the test tube. So what Meselson and Stahl did was they took advantage of the fact that nitrogen came in heavy and lighter forms. So what they did is they started out with a bunch of E. coli that had grown up in heavy nitrogen, and then they transferred it to some lighter nitrogen for one generation, and then they transferred it one more time to light nitrogen for generation 2. Now let's look at their results. Once they centrifuged down their samples, here's what they found. So this is our first sample. These are the E. coli that all grew in heavy nitrogen solution, okay? So I just wanted to point that out. So you can see that we've centrifuged this test tube, and this little dark spot right here, this is the radioactive part of the nitrogen, okay, that you're seeing. This radioactivity made a stain on some photographic film, and that's how we got this image. So basically what they did was they took a test tube, they centrifuged down their DNA, and then they exposed it to some photographic film. And anywhere that there was N15, anywhere that there was radioactivity, it made a line, okay? So let's look at these results really closely. Round 1, all of our DNA has heavy nitrogen in it. So here is our line, okay? So I'm gonna draw that in black. This is all, if it's black, it's gonna be heavy nitrogen DNA. They let that go on for one generation. Then they moved their E. coli to a new growth medium that had light, it had N14 in it, okay? And they let it grow for a little bit, and then they centrifuged it down. And I want you to notice something. Do you notice that this line is higher up than this line? That is because this DNA was lighter than this DNA. Now, why would that be? Well, if semi-conservative replication is true, then what happens is you have one heavy strand of DNA, like that, and like that. And then after you've replicated it, you're also going to end up with a light strand of DNA. And so together, this molecule, because it's got some light nitrogen and some heavy nitrogen, it's a little bit lighter than this molecule. Same with this one. Okay, this is our light nitrogen. This is our heavy nitrogen. But together, these two strands are a little bit lighter than these two strands. And that's what accounts for this line being higher up than this one. Now, they went and let this go on. They continued to let these grow in light nitrogen solution. And they, centrifuged the DNA one more time. And what did they get? Well, they got 2 lines this time, okay? One of the lines was right here. These are of equal size, and then one was even higher up. How can we explain that? Well, let's take these 2 guys, okay? And let's separate them out again. So let's start with this one here, all right? I'm gonna move him down here. So this is just me copying that strand over that's got one strand with heavy nitrogen and one strand with light nitrogen, okay? So this is our intermediate weight. Now, if we pull those apart, So I'm going to pull that apart. Boom, 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 boom on one side. And I'm going to pull this one apart. And I told you that we were still growing things up in light nitrogen. So all we have is light nitrogen nucleotides. Look here. This molecule of DNA is going to weigh the same as this molecule of DNA here, isn't it? So look, this and this are at the same place. They're the same weight. But this molecule of DNA has no heavy nitrogen at all. It is just N14 and N14. And so this is even lighter up here, okay? So make sure that you understand this experiment. If you don't, there are a number of YouTube videos that will explain this line of reasoning to you, but I just really want you to understand the thinking here and to understand that this experiment, the Michelson-Stahl experiment, demonstrated that DNA is replicated in a semi-conservative fashion. Okay? And I think I'm going to stop this here, and I'll actually do one more part of this video so that we can kind of break this into chunks. In our next part, we're going to learn exactly how this process of replication occurs. Talk to you soon. Bye-bye.